Hello students, I am Dr. Aruna Mohan from University of Delhi. Today in this session we will discuss about sense organs. You know, we have 5 important sense organs in our body: eye, ear, nose, tongue and skin. Eye is a sense organ for vision and is very important. We all know how important eyes are for us and how interesting it is for us to see various things around us. Suppose we did not have the eye, then world will be so different for us. We enjoy each and everything around us because we can perceive, we can see. So today we are going to discuss how our eye is and how it functions. Eye is placed in a round structure called eyeball, and within this eyeball the eye is well protected. As far as structure is concerned, it has 3 major layers. The outermost is called sclera or scleroid. It is important because it is going to make cornea towards the anterior part of the eye. The middle layer is choroid, which extends and makes ciliary body, and this ciliary body will further extend and make iris, and this iris will accommodate crystalline lens. Lens is almost a non-living substance, and this lens is surrounding one small aperture which is called pupil. So, these all structures are interrelated and their functioning is controlled by muscles which belong to eye, or we can call them eye muscles. You can also see the third layer we call retina. I am showing it in the diagram. This is retina inside one, and it has got ganglion cells. The moment I use the word ganglion, I am meaning the nerve cell. These ganglion cells are bipolar cells. I would like to explain the term bipolar one more time. You remember the structure of nerve cell. It has dendrites, it has one axon, and if it is having no dendrite, only axon, we will call it unipolar, means one projection. And if it has one dendrite and one axon, we call it bipolar, and if it has many dendrites and one axon, we call it multipolar. So, 2 things come out of it. First thing, axon is always there. Dendrite can be either it is not there or can be 1, 2 or many. Depending upon the projections, we define the nerve cell. So, if I say bipolar, I mean one dendrite and one axon. In your eye, the kind of nerve cells present in retina are bipolar, having one dendrite and of course one axon. This is a special point to remember in this context. Also, there are photoreceptors. These photoreceptor cells are rods and cones, which have specific features for the vision, the color vision, the daylight vision, the twilight vision, etc. In this diagram, you can see this outermost is sclera, the middle is choroid and innermost is retina. Also, you can see the cornea, you can see pupil, you can also see lens and the iris. So, this is the placement of various structures in our eye. You all have idea about vision. When we see thing, any item in daylight or in artificial light or in twilight or in trying to see in a dim light, things look little different because our eye will accommodate differently. So, vision becomes very important, and it is very important for us to know how all this is happening in our eye, which is a very sensitive organ. The daylight vision and the color vision is looked after by cones, whereas twilight vision is looked after by rods. These rods and cones are photoreceptors, and they have photopigments which are going to help in the vision. Let me also tell you something about optic nerve. I have told you about retina. This retina will finally go towards brain in the form of optic nerve. In this particular slide and the diagram, you can see the part of retina. This is part of retina, and this is a point from where retina is going as optic nerve, and finally it will reach cerebral cortex. Also, you can see the point from where it is making an entry towards the brain or exit from eyeball. At this point, we do not have rods or cones. That means it is a blind spot through which the optic nerve is travelling out. Near this blind spot only you will find another spot which we call yellow spot. This yellow spot has central pit called fovea, and that is a point where image will be formed, and it will help us in seeing the objects. Now children, let us also understand about certain chambers in our eye. We have two major chambers, one between cornea and the lens, we call it aqueous chamber, and one between cornea and retina, we call it vitreous chamber. The aqueous chamber is filled with watery fluid, we call it aqueous fluid or aqueous humor, and the vitreous chamber is filled with jelly like substance, we call it vitreous humor. Both the chambers are significant, and these fluids, they provide support for the size and the shape of the eyeball. I will now explain it in the diagram. I am showing you in the diagram the chambers. This is cornea, you can see, and here is the lens, and between cornea and lens this is aqueous chamber which is filled with aqueous humor. And now I will show you vitreous chamber. This is lens and this is retina. This whole thing is vitreous chamber which is filled with vitreous humor, which is gel like substance. So, these two chambers which are filled with fluid like substance, the watery fluid and the gel like fluid, they are responsible to keep this eyeball in its shape. So, it remains in its shape and does not sink inside. Now, we will understand the mechanism of vision, how we see any item in front of our eyes. Light will fall on retina. We all know what retina is, and when light falls on retina, please remember what I explained to you in previous session about action potential and reflecting potential, because in this case when light falls on retina, those kind of changes will take place. So, it will generate potential in rods and cones. I told you that rods and cones are photoreceptor cells and they have photopigments. Those pigments are also very important to see different colors in different intensities. So, when light falls on retina, it will generate potential in rods and cones. It will affect photopigments which are called opsin and retinal. So, these photopigments are affected by change in the potential which was created due to falling of light on retina. Due to all these reasons, the membrane potential will change. You all know what I mean by potential. And once that happens, the potential is generated. and when that is generated, that will produce signal that generates action potential in the ganglion. We have talked about ganglion in the retina. Once the action potential is generated, the work is done. You know that nerve cell or the membrane is in a resting condition originally, which is resting potential. And when gradient changes, when potential changes, then it comes in the action potential mode. That means it has come into the action mode. Now, this change in the action potential, it is carried by optic nerve to the cortex of the brain, especially the cerebral cortex, and more specifically the center for vision. So, students, you have understood one thing: when light falls on retina, it creates change in the potential, and when resting potential is converted to action potential because membrane was affected, at that point of time the potential which has changed is carried on by optic nerve from eye to the cerebral cortex of the brain, more specifically the area for vision or center for vision, where we are going to see the image. This is how the things really happen. You are trying to see one ice cream cone. This you want to see. Now, light rays from this will cross the lens and will fall on retina, where? On the yellow spot, because that is a spot where we have photosensitive cells, rods and cones. Please remember, the light rays are crossing each other, so image is going to be inverted. And when optic nerve takes this image to the brain, again there will be crossing of the light rays, and again it will become straight, erect. So, you will see the erect image, not the inverted image. Children, you may be remembering when you were a small child, you must be making whole camera with a shoebox and trying to see some object in front of you, and you will find the inverted image. That is the thing which is happening up to the retina. After that, it is made erect in the brain because it is taking one more round, the rays are crossing one more time. So, thing to remember here is the light rays will pass through lens. So, lens is important, the illumination outside is important, an inverted image to be formed at yellow spot is important. Suppose yellow spot is not at the correct place, then image will not form. Those are the eye defects sometimes, and it is again becoming erect in the brain. Now, how much light should go inside? That is decided by our iris due to the movement of eye muscles. When you are in the daylight and suddenly you come inside the room, you are not able to see things for some time, and then after some time you are able to see, because your eye will accommodate the pupil, the lens through eye muscles. So, eye muscles are also very important. The illumination outside is also important. So, only when light rays enter the crystalline lens, you can see the image. So, this is the mechanism of vision, how our eye, if it is normal eye, is able to see. Suppose yellow spot is at a different point, then accommodation will be different, and then person has to put aid or chashma, which will adjust the light rays and the spot where image should be formed. So, in today's session, we have understood how our eye works and what are different structures of our eye, and also we have understood how important the functioning of eye or this sense organ is for our body. Thank you.